Good evening, everyone. Today, we'll be starting with the discussion of week 4 for the RNA biology lectures. I'm Rishabh, and I'll be starting with the lecture. And in the last class, we started with that RNA can be a catalytic molecule, and like proteins, RNA can also have some catalytic role as per the RNA world hypothesis. So, as per this hypothesis, as we have discussed earlier, RNA can have dual roles. That is, first role is the self-replicating molecule, and the other role is catalysis. Two major role involved with the functioning of the life. So, what is basically the evidences that we also discussed about the evidences for RNA having a catalytic role. So, we discussed that the discovery of the catalytic RNA, that is called as the ribozymes, that were done in late 1980s, and that was basically discovered by the discovery of many catalytic RNAs. One example is ribosomes. So ribosomes are large ribonucleoprotein complexes that helps in translation of RNA into proteins. Now, ribozymes are also artificially created RNA molecules that can catalyze any reaction outside the body also. We'll discuss with these applications of ribozyme and how these helps in any of these catalytic roles outside the body. And then these RNA sequences are capable of simple forms of self-replication also. So this is in cooperation with the RNA world hypothesis. So basically, the ribozyme means that RNA that have a catalytic role or that behaves as a catalyst. Okay, so this is an enzyme, and the biochemical nature is that it is a ribonucleic acid, and but it is not a protein. So this in 1989, the discovery of ribozyme by Sid Altman and Tom Say uh, received a Nobel Prize in chemistry. So for the discovery of RNA in living cells that is not a molecule of heredity, but it is a molecule of biocatalysis. Okay, so this is the Nobel Prize winning discovery of the ribosome. Now, followed by this, now we also discussed that the structure plays a main role in developing catalysis function in RNA. So, what happens as proteins also have, so if we talk about proteins, proteins also have a primary, secondary, tertiary, and quaternary structure. So, talking about the primary structure, we have the protein sequence or the amino acid sequence. Then, after this, the proteins fold upon itself, forming two basic units called as beta sheets or alpha helices. And there are multiple secondary structures that we're not talking about right now, but we discussed also that folding of the amino acid chain upon itself forms either of the secondary structures in protein. Now, followed by this, these structures fold upon itself and form a motif or fold kind of structure that have a functional role. So this particular functional role depends upon the conformation of the folding of these secondary structures upon itself. So this forms the tertiary structure of the protein that provides the function of the protein. Now, followed by this, if there are multiple subunits and multiple components of a protein, they get folded and form a quaternary structure. That also provides the functional role if it's a multi-subunit protein to the functional role of that protein. Okay, so this particular structure provides the functional role of the multi-subunit proteins. Similarly, if we talk about RNAs also, it also have a primary structure. So the primary structure of RNA, being a nucleic acid, it will have only the sequence of A, U, C, and G. So the continuous sequence of these four nitrogenous bases will form the primary structure of the RNA. Following this, because there would be some complementary base pairing among the nucleotide sequence itself, that will fold upon itself and form the secondary structure, like formed followed by this. Okay, so now this has some complementary base pairing, and it will form a functional fold. Okay, now followed by this, it will fold upon itself due to various other interactions of van der Waals and electrostatic interactions. It will fold upon itself and form the tertiary structure of the RNA. Okay, so this will also have a specific fold, or this specific fold that will provide the functional role to the RNA. Okay, so this tells us that RNA also has these kinds of structure that will give a catalytic functional role to the molecule. Okay, so now here we compare the structure of the ribozyme and the tRNA. Okay, so as we also discussed that tRNA also have a secondary and a tertiary structure. So talking about its secondary structure, it has a clover leaf kind of structure which forms stems and loops. As similar is the case with the ribozyme. It also have certain stems and loops that provides or helps in forming the catalytic subunit of the ribosome. Now, similarly, for if it folds upon itself, that forms the tertiary structure. This is the L-shaped structure of the tRNA. Okay, and this is the complete structure of the ribozyme after folding. This basically folds, forms the catalytic role or the catalytic site of the ribozyme where it performs its function. So moving on, so as we all also saw that the protein also has a, like, in case of protein or enzyme that we see generally, we have a active site, that is the actual site where the reaction occurs. Okay, so where the acid base catalysis or the binding of the substrate to the protein happens, and the protein and the substrate is basically converted to the product. So this particular active site is the functional part of the enzyme. And in ribozyme also, there is one active site that is formed of different bases due to the folding upon itself. Okay, so here is an example of, like, in proteins we have different amino acids present in the active site. So taking an example of chymotrypsin, so this is the molecule of chymotrypsin. And here, as we saw that in the charge relay system, so this is the protein molecule that needs to be cleaved off, and chymotrypsin is a peptidase. So here the three molecules, serine, histidine, and aspartate, are the catalytic triad present in the active site of these proteins. So these proteins helps in modifying, attacking the protein through various nucleophilic attacks, and modifying the proteins to the forming the product or the cleaved protein. Okay, and here the glycine residue helps in positioning of the protein so that these three molecules can work together and do its function. So like this, the purpose of taking this example is to show that the RNA ribozyme also have an catalytic site that helps in doing the cleavage or the function of the basic ribosome. Okay, so now we'll be moving on to the naturally occurring ribozymes that present in our body. Okay, so I'm giving certain examples and what functions they perform. So first is the rRNA. So as I told you before also that in ribosome, these molecules are present specifically if we talk about the peptidase transferases. So 23S rRNA present in the ribosomes of prokaryotes, and 28S plus 5.8S rRNA present in the ribosomes of the eukaryotes. They both function as peptidase transferases. Okay, so these both are the ribozymes because they have a catalytic function and they help in peptide bond formation. So they basically help in the protein synthesis. Now, the other example is ribonuclease P, or in short, we will say it as RNase-P. So it helps in hydrolysis of the phosphodiester bond, that is between two bases. So these bases are joined together forming a polynucleotide through phosphodiester bond. And this phosphodiester bond is broken by can be broken by a RNA enzyme or the ribozyme RNase P. And this RNase P helps in maturation of the tRNA. Okay, so later when we'll see the function of ribosome P, as well as when the tRNA formation that we'll see, we will see the RNase P and how it is involved in the maturation of the tRNA, forming from precursor tRNA to the mature tRNA. Okay, now then we have cell splicing group 1 and group 2 introns, and these also have the role in phosphodiester bond cleavage as well as ligation. Okay, so these self cleave themselves out of the exons and they'll ligate the exons. Okay, so they have a biological role in removal of the introns from the precursor mRNA. Then there comes the hairpin ribozyme or hammerhead HDV. So these also have a role in phosphodiester bond hydrolysis, and they basically helps in replication or in the infectivity of the viruses, RNA viruses. Okay, especially hepatitis delta virus that we have here. So the detail we will see later. And these are basically the examples of ribozymes that are present in our living body and functions like the normal protein enzymes. Okay, so some of the questions are asked in various competitive examinations, like this ribonuclease P is involved in various like functions in the body, and what are those? Similarly, for the self splicing group 1 and group 2 introns, and so these type of questions are also asked in various examinations. So moving on, there is less, like I told before also in the previous slides, that there will be some similarity from the from the ribozyme to the protein enzymes. So like both of them are enzymes, so there will be some similarity between both of them. So we'll talk about this particular aspect in this slide. So what happens, like in ribozymes and protein enzymes, the acid base catalysis and metal ions activate the nucleophile group to stabilize the developing charge on the leaving group. So both of them uses, for example, this is a particular ribozyme and it forms a functional fold. Then this is the active site of the ribozyme, and this is the protein molecule, and this forms the functional fold, and it both requires a cofactor Mg2 plus. Okay, so this Mg2 plus helps in the activation of the nucleophile groups present at the active site of the ribozyme. So this is the ribozyme and this is the protein enzyme. So like Mg2 plus, it will act upon the bases present in the active site, and it will activate those bases so that they can attack to the substrate. And likewise, Mg2 plus in protein enzyme activates the amino acid side chains. Okay, so for example, the chymotrypsin example that we saw, and other protein enzymes like lysozyme. So what happens, these basically the cofactors or the metal ions, these activate the side chains of the amino acids, so that will act upon the substrate. Okay, so and there are various acid-base catalysis where the phosphate group. In case of the nucleic acids, the phosphate group performs the acid-base catalysis because the nitrogenous bases do not have any form of charge on them. They are only the nucleophiles. They act as a nucleophile because they have a, like for example, the in adenine, when we see, we have a NH2 group that will have a lone pair. So this is the extra lone pair present on the nitrogen, so it will have act as a nucleophile. Okay, so similarly, when we talk about the protein enzymes, they have negatively charged amino acids like glutamate or the aspartate, and positively charged amino acids like histidine, uh, lysine, and then we have aspa- uh, and that we have arginine. So these performs the acid-base catalysis by donating or electrolysis form of an interaction. So these are the similarities that have the between the ribozyme and the protein enzyme. Then, as I said, that ribozyme also requires a formation of the specific tertiary structure that will give it a functional fold. And then, like enzymes, also increases the rate of reaction by providing the environment that can convert a substrate into product. The ribozymes, for example, hammerhead RNA and others, also are known to be increase the rate of the reaction so that the product formed is higher as it compared to the product when it is formed in another way. Okay, so yeah, like HDV ribozyme or the hepatitis delta virus ribozyme cleaves the phosphodiester bond as fast as RNA. So it also increases the rate of the reaction as like in the protein enzyme. Okay, so these were the certain similarities that we see between the ribozyme and the protein enzyme. Okay, so we'll be discussing more about the types of ribozymes and the details of some of the ribozymes that we see in our human body. So, depending upon the structure of the ribozymes, okay, so this classification is based on structure and the use of metal ions for catalysis. So, if the ribozyme uses metal ion for catalysis, so these are called as metalloribozymes because they use metal ions as cofactors for catalysis. Okay, and then we have some small ribozymes that basically helps in RNA cleavage and have certain applications that we'll see in the end. So these molecules basically helps in RNA cleavage, and they do not have any metal and as cofactors. Okay, so in the metalloribozymes we have two kinds that we'll discuss in detail. That is ribonuclease P, or as RNase P, and the self-splicing introns, or the group 1 and group 2 introns. And in small ribozymes we'll be discussing about the hammerhead ribozymes, in hammerhead ribozymes, hairpin ribozymes, and hepatitis G virus ribozymes. Okay, and then in the last we'll discuss about the peptidyl transferase that do not fall in any of these of the classes, because its function is very different from these two classes, and how it helps in peptide bond formation. Okay, so let's start with the metalloribozymes and the RNase P. So first molecule is RNase P. So as the name suggests, this is RNase P, ribonuclease P. It will cleave the RNA. So it will cleave the RNA. So the substrate would be RNA only. Okay, so this RNase P is involved in the maturation of the tRNA, as we have seen before. Okay, so major function, or if we talk in detail, so it will cleave the 5 prime leader of the precursor tRNA, so that we can generate the mature tRNA, or the mature 5 prime end as a prerequisite for the tRNA function. Okay, so like give you a glimpse of where the RNAsP act, and we'll see the major process of maturation of tRNA when we'll discuss about the splicing mechanism. So what happens during tRNA formation? This is the clover leaf form. So this is the 3 prime end, and this is the 5 prime end. Okay, so this is clover leaf structure of the tRNA. So this 5 prime has a leader sequence. Okay, so this sequence is not needed in the mature tRNA. So RNase P cleaves this particular part, and this is removed, leading to the uh, tRNA flanking the 3 prime end. And later on, this tRNA also get a CAA trinucleotide repeat at the 3 prime end, forming the acceptor arm of the tRNA. Okay, so here is the role of the RNAsP, removing the 5 prime leader sequence in the tRNA. Okay, so now we'll move on to the structure. This was the function of the RNAsP, and how it helps in maturation. Now talking about the structure of RNAsP, that we'll discuss a bit in detail, but to start with, it has a RNA component, obviously, because it has a catalytic RNA unit, and it is linked to a protein that is called as a P protein. So the RNA component is called as the pRNA, because as the name suggests, and the protein component is called as the P protein. So RNase P will be a ribonucleoprotein. Okay, so if anyone asks you that what will be the biochemical composition of the RNase P, so the answer would be that it has a RNA component as well as the protein component. So it will be called as a ribonucleoprotein. Okay, but the major catalytic function is due to the RNA component only, and not the protein component. So the catalytic unit is formed from the RNA component only, not the protein component. And it is present in all domains of life, being it prokaryote, eukaryote, or archaea. Sorry. So, as we can see in the following image, that first we'll talk about the simplest form, that is bacteria. So, we have this is the RNA component, and there is only one protein involved. Okay. In the archaea, it is the RNA component, and there are five proteins involved. In the eukaryotes, there are different forms of RNAs. So this is RNase P, this is RNase MRP. So what happens? There are 10 to 12 proteins involved. So depending upon the complexity, the number of proteins increases that works with the RNase molecule. So the more the complex organism, the more the proteins involved with the RNAs. So talking about the protein component, it increases with the complexity. So first comes the prokaryotes, then archaea, then comes the eukaryotes. This is based on the number of proteins involved, or that is linked with the RNase P molecule. Okay, so this is the basic information about the RNase-P molecule and where it is involved in function. Now, talking about a bit about its structure. So this is basically the 3D structure of the RNase-P molecule, and if we see this, we have talking about the pRNA first. It has two domains. One is the S domain and other is the C domain. So S domain stands for the specificity domain and C domain stands for the catalytic domain. Now the RNA component is around 338 nucleotides. So this we are talking about the E. coli, the simplest and the first RNA speed that was to be crystallized and its 3D structure was visualized. So it has a 338 nucleotide long RNA and the protein component is nearly 120 kilodalton. Okay, and uh, the protein component is basically joined with the uh, to the catalytic domain where the the leader sequence or the 5' leader sequence of the tRNA gets cleaved and it needs to be removed from the tRNA as well as the ribozyme or the RNAsP. So this protein is linked to the catalytic unit, and this specificity domain helps in binding of RNase molecule to the tRNA. Okay, so how do RNase bind to tRNA? Through complementary base pairing. Okay, so this is the basic structure. So this the protein component is outside the catalytic domain, just linked to the catalytic domain, and where this 5' end. So this protein component, what is the function? So it has basically two functions as mentioned in the literature. First is it carries the 5' leader sequence to uh, that basically that is removed from the tRNA and released. Okay, and the second component is it basically helps protecting the RNase molecule through various nucleases because it's a RNA only, so it is more prone to attack by the nucleases. So it basically protects the protein component protects the RNA from the attack of the nucleases. Okay, and you can see this is the tRNA molecule, and here is the zoomed in image of the 5' leader sequence, and this is being cleaved by the RNase P molecule. Okay, and this is the 3' CCA that will be added afterwards. Okay, so this is the how the structure, and it helps in attaching the ribonuclease molecule to the tRNA. Okay, now moving on in a bit deep into the mechanism and how it helps in uh, removing uh, the 5' leader sequence. Okay, so what happens? This is the this is the tertiary structure of the tRNA. So this shows the primary recognition for RNase P. So this acceptor stem recognized by the RNase P molecule like this. Okay, and here because it's a metalloribozyme, so Mg2 plus also plays a major role uh, in degenerating or in hydrolysis of the phosphodiester bond. So how it happens? So this is the base at the -1 position of the pre-tRNA. Okay, and this is the leader sequence of where the cleavage will happen. So now magnesium ion comes and it interacts with the nucleophile groups of the basic sugar as well as the phosphate group. Okay, that will only help in cleaving the phosphodiester bond. So now what will happen here? It interacts with the 3' OH group. It interacts with the 3' OH group of the sugar molecule that is the base that needs to be cleaved. Okay, so and the nucleotide that needs to be cleaved. So it interacts with the 3' OH molecule through water molecule, and it interacts with the phosphate group, okay, of the other molecule. So this is the sorry it interacts with the 2' molecule, yeah, sorry, that it interacts with the 2' molecule of the base of the leader sequence from which it needs to be cleaved, and the 3' 5' phosphate of the previous molecules to break off the phosphodiester bond. So this basically activates the nucleophilic basis of these phosphate and the OH group using magnesium ions. So what will happen? This creates tension on this particular oxygen group at the 3' end, and this bond breaks and it gives the electrons to the oxygen group. Okay, so what will happen? This is the 5' leader, and here what will happen? This oxygen will have the both the O molecules, and this is the OH group forming a hydrogen bond with the H2O molecule. Okay, and here the phosphate group uh, also has one electron. Now what will happen, water will come into play and it will provide hydrogen as well as OH group to the phosphate group. Okay, so magnesium holds this water molecule and it uh, now this is the RNA chain. So now what will happen? The 5 prime leader is released, and here it is left with the 3 prime single stranded RNA. That is the acceptor arm of the tRNA, mature tRNA. Okay, so this is how magnesium plays a role in the in activating the nucleophiles of this tRNA so that the cleavage can happen. Okay, so in like briefly, magnesium ions activates water that targets the phosphodiester bond. So this is the basic pathway of or the mechanism of how the RNAsP works. Okay, so if you have any questions, you can ask. So this completes the RNAsP molecule, and we'll be moving on to the next molecule, that is the self-splicing introns. Okay, so these introns are basically greater than 200 nucleotide long, or rather 200 ribonucleotide to be specific. Okay, so these can splice themselves out. This is the function of these particular introns, and they do not require any protein to help them out. So they are self-sufficient and they can cleave out from the RNA chain. These introns are found in both prokaryotes as well as eukaryotes, and these like to give specific examples, these are present in the primary transcript of mitochondrial genes of yeast and plants and pre-RNA of the tetrahymena. This is a protozoan. Okay, so this has been discovered till now. So these RNAs are present in the primary transcripts of the mitochondrial genes of yeast and plants and the pre-RNA of the tetrahymena. That is a protozoan. Okay, so we divide these two introns into group 1 and group 2 introns based on their mechanisms and how they cleave out, that we'll discuss just after this slide. And so group 1 introns are found in some of the nuclear, mitochondrial and chloroplast genes that encode for rRNAs, mRNAs and tRNAs. Okay, so all the three major RNAs genes have these in some of the genes have group 1 introns. Okay, so this is present in nucleus, mitochondria as well as chloroplast. Talking about the group 2 introns, so these are found in the primary transcripts of the mitochondrial and chloroplast mRNA in fungi, algae as well as in plants. Okay, so obviously these are introns, so these will be present in the primary transcript only. Okay, so starting with, this was the introduction of these particular self-splicing introns. So talking about it, these introns involves a particular type of reaction called as esterification. Okay, so this is basically involves a formation of the phosphodiester bond between different molecules forming a cyclic group, and this is called as a transesterification reaction. This is a basic feature of the mechanism of these particular introns. Okay, so first we'll discuss about the transesterification. Before that, let me give you a brief that I would have given before also. So, talking about the prokaryotes, if we see, the prokaryotes have a gene sequence starting with the promoter, and then the major unit, and then the terminator sequence. So this full length is the protein coding gene. So this is called as the open reading frame. Okay, and here there is no intron present. Okay, so the full length is the cistron, or that codes for a protein. Okay, and in eukaryotes, if we see, we have promoter, then we have alternating sequence of the intron and the exon. Okay, so what happens? The major protein is coded by the series of exons, and introns are the ones that are removed during a process called as splicing. Okay, so followed by the terminator. Okay, so here the introns are removed and the exons are formed, forming the exons are joined together, forming a open reading frame that will form the protein via translation. So this is the example of the eukaryotes. So this place splicing reaction basically is more involved in eukaryotes as compared to the prokaryotes. The prokaryotes also has some of the group 1 and group 2 introns that basically involved in the cleavage of different RNA molecules. Okay, so now um, first I'll erase this. So this was to basically introduce to those people who don't know about the gene structure and the prokaryotes and eukaryotes, where the introns are involved and why we are studying splicing reactions. So here we'll be studying about the transesterification, that is a feature of the self-splicing introns. Okay, so now what happens? This is the structure of the exon. So this is in green color, and in the yellow color we have the intron. Okay, so now what happens? There will be any nucleotide or other base that involves in the nucleophilic attack. Okay, so generally the 3' OH is involved in the bond formation through the next next base. So any of these sorry so the this is the 3' OH, this is the 2' OH. So the 3' OH is involved in the bond with the next base through 5' 3' phosphodiester bond. Okay, So now what happens? Another base comes and it attacks the phosphate group through a nucleophilic attack. So this is basically the 2' OH because it's free, so it's catalytic in nature. That is what we studied till now. So what happens? This 2' OH attacks the phosphate group because now as the bond is formed, it is in a state of like what we call in chemistry as electron deficient state. So it acquires a particular positive charge, and then this particular the electrons attacks this phosphate group, and what happens? This phosphate group now donate or links with this particular oxygen molecule, that is the 2' OH group. Okay, so now this bond is broken and the electrons involved in this and this bond are shifted towards the oxygen molecule. Okay, so this leads to the breakage of this particular bond with this particular nucleotide. Okay, and this is a new phosphodiester bond formed between the 2' OH of this particular base and the 5' phosphate group. So this is a phosphodiester bond only like this, and phosphoester bond, and this is 2' 5' phosphoester bond. Okay, so now the ester bond between these particular bases, that is 3' to 5' is shifted to 2' 5' 2' 5'. Okay, so this process of shifting of the ester bond from the 2 3' to 5' to 2' 5' is called as transesterification reaction. Okay, so this transfer esterification reaction is the basic feature or the mechanism involved in cleavage of group 1 and group 2 introns. Okay, so if you have any doubt, you can ask. So moving on to the group 1 introns and its splicing mechanisms. So what happens? This is a primary transcript where there is an exon. Between these two exons, there is an intron. So for simplicity, we are taking only one intron here, and that have a 5' exon and a 3' exon. Okay. So considering this, we have a U at the 5' end of the first exon and U at the 3' end, oh sorry, 5' end of the 3' exon. Okay, so what happens in case of group 1 splicing mechanism? The G or the ribonucleotide guanidinate. Okay, so GTP basically has a major role. So guanosine or any of these uh, forms of GMP, GTP, and GDP. Okay, any of these can help in the removal of this particular intron. So the nucleophilic group that we talked about just now is provided with the 2' OH of this particular uh, guanosine. Okay, so it attacks any guanosine present in the nucleus. So it attacks the phosphate group of the linking uh, intron-exon phosphodiester bond. So it attacks in a nucleophilic way. And what happens? The bond is broken between the U and A, and this particular exon is formed. Okay, and Following the trans-esterification reaction, this OH 3' OH is free. Now this 3' OH also acts as a nucleophile, and it attacks on the junctional phosphate group of this particular intron and exon, thus cleaving this bond, and this particular structure is released. This is the intron part, and this is the this is joined, forming the complete ORN. So this is the spliced RNA or the mRNA, mature RNA, that will go for translation. Okay, so this is how the group 1 intron is self splice itself from a primary transcript. So here, the external guanine nucleotide attacks on the phosphate group of the junction. It cleaves this particular phosphodiester bond. Then the 3' OH, the empty OH of the exon 1, targets this particular uh, junction of the intron exon, and it cleaves this particular bond and relieves the intron. Okay, and this is combined, leading to formation of the spliced exon, sorry, spliced RNA or the mature RNA. Okay, so this was the basic mechanism of the group 1 self-splicing introns. So it also have a certain role. So after splicing, these introns can behave as enzymes. So this also was a ribozyme kind of thing. But now these RNAs will also behave as a ribozyme and help other RNAs or cleavage of other RNA molecules via internal guide sequence. So what happens? Like for example, this particular intron is cleaved from the uh, primary RNA. Okay. Now it has a internal guide sequence that is complementary to the RNA of other protein molecule. Okay, so it will directly target this RNA, bind to it because it has a complementary base pairing, and it will cleave this RNA. Okay, so the protein will not be formed. This was the application that is currently being used by different scientists to develop treatment for various viral infections, and this was the first point. And second, treatment for various diseases like cancer. Okay, so what we can do is, we have the internal guide sequence. Okay, so we can change this internal guide sequence to different target molecules involving, for example, if we have the viral RNA sequence of one viral RNA, so we can directly this will directly target this viral RNA, and it will cleave those RNA, leading to the loss of infectivity of that particular RNA molecule. Okay, so this is how we like the scientists and the clinicians are using this particular mechanism for the treatment of various diseases, especially who do not have any cure right now. Okay, so this was the group 1 intron and how we can use it for the application, different applications. Now, moving to the group 2 introns. Now, starting with the group 2 introns and its splicing mechanism. So, it is a bit similar to the group 1 introns, but here the attacking group is the molecule present within the intron itself. Okay, so here also the same example. This is the 5 prime exon, this is the intron, and this is the 3 prime exon. So here we have a point that is at the branch point. Okay, this is called as the branch point. And here, this branch point A attacks as a 2' OH that will attack the junctional phosphodiester bond of the exon intron, and this cleaves this particular bond. And the second thing what happens is the cleaved bond will have a 3' OH, that is a free 3' OH with a lone pair. So this will attack the junctional phosphodiester bond of this exon intron. Okay, so now what will happen? This will cleave, this phosphodiester bond will cleave, leading to a, a removal of the intron that is a complex structure. This is called as the lariat structure, which, which have both 2 prime, 5 prime, and so as you can see in this particular case, this A has a 2 prime 5 prime phosphodiester bond with the G molecule, a 3 prime 5 it bonds with the mm, 3 prime 5 prime with this A molecule, and 2 prime 5 prime with this particular C molecule. So this A has 3 phosphodiester bond with these particular nitrogenous bases that forms a lariat structure or a curved structure. Okay, and this is a complex structure basically, and that will be cleaved further with the nucleases present inside the cell, and this will lead to formation of the spliced RNA or the mature RNA. Okay, so this is basically the group 2 introns. And their splicing mechanism. So here, no external protein is involved. So protein is not involved. And like the group 1 introns, the external G nucleoside is also not involved. Here, the nucleophilic attack is done by the is done by the this nucleoside, the nucleotide base of the branch point, or that is present inside the intron itself. Okay, so now this is the thing, and now how these are in present in the metalloribozymes. So here also magnesium ion, as we have seen in case of the uh, ribozyme structure, this particular, that how Mg2 plus helps in the breakage of the and the nucleophilic attack of this particular nitrogenous base. So these also have a magnesium ion involved in the breakage of the phosphodiester bond. So that is why these are called as the metalloribozymes. Okay, so now I have a small question for you guys that we'll discuss in the next lecture. It although, but how will you design an experiment to check for the presence of group one introns, or just like think about it that how group one introns were discovered, how scientists would have thought that there could be some kind of mechanisms like this. Okay, so you just like pull up for the techniques what which we can use in identifying the group 1 introns or any other this kind of ribozymes. But here we are discussing specifically about the group 1 introns. Okay, so you just think about it and we'll discuss in the next lecture. So now moving on to the small ribozymes, that is the hairpin ribozyme. So what happens in this case? So like as we have discussed before, that when RNA molecule have a folded structure, so in the terms of RNA, the fold is in the form of loop and stems. Okay, so like this is the primary structure of the RNA and it forms different stems and loops. Okay, so here is the stem of the RNA molecule, the ribozyme molecule. This is the loop, and here different structure it forms the catalytic site. Okay, so this is the basic structure or the loops that are formed during these structures. So these RNA motifs, the or the hairpin ribosome that we are talking about, these motifs that catalyze RNA processing reactions or involved in the replication of the satellite RNA in which they are embedded. So this hairpin ribosome, so hairpin is this kind of structure that is formed. Okay, so these are basically involved in RNA processing and RNA replication. So this was first identified in satellite RNA of tobacco ring spot virus. Okay, so these are like discovered or found in these particular tobacco ring virus. So what happens, this is the substrate for this particular so this is the structure of this particular RNA, and what it does is this has a this kind of structure. So till here is the structure, and in this it forms a loop. This is the 5 3 prime end free, and this is the 5 prime end of the ribosome. And what it does is this particular G8 is the main attacking group, and it attacks on the this particular phosphodiester bond of the target RNA on the substrate. Okay, and this arrow shows the cleavage site. So as you can see, this and this forms the complementary base pairing or helps in binding. Okay, so here the black, so that means if this particular RNA is targeting a specific RNA, so it must have the same sequence or the complementary sequence at least, so that it will target the specific RNA. So that is why all the bases mentioned in the black are variable. It will change from different viruses, and these blue ones are semi-conserved. Okay, so that can be present in some of the viruses but not in the others. And this orange ones are highly conserved. For example, this G8, that forms the cleavage site or the catalytic center of that particular ribosome. So it targets the substrate RNA. Okay, so this was the basic structure of the hairpin ribosome found in the tobacco ring spot virus. So now, what is the main mechanism and how it functions? So this is the mechanism that we are talking about, and this is the process that it how it helps in cleaving the virus. So what it does is, so this is the structure of the ribosome, this one, and here, sorry. So Here, this is the target RNA that needs to be cleaved. First, what happens? It binds to this. This is called as docking. So it docks, or it identifies the target RNA and binds to it through complementary base pairing, and then the G present in the catalytic site gets activated and it targets the target RNA and cleaves it. So the next step is cleavage, followed by the undocking, or it leaves that particular RNA like this. So it releases the RNA. And this is the basic mechanism of how the G8 that is involved. So this is the G8 molecule, and here the N, or the nitrogenous bases present in the ring of the particular purine nucleotide, targets the 2' OH. Okay, and this 2' OH basically this 2' OH targets the phosphodiester bond and cleaves it, followed by the cleavage of the particular RNA molecule. Okay, so this is how the basic mechanism remains the same. The nucleotide or the any other molecule that have a nucleophile group targets the phosphodiester bond, and that phosphodiester bond is broken down, forming the free 5' and 3' ends and relieving the two parts. Okay, so this is the basic mechanism, and this remains constant throughout the cleavage patterns of different ribozymes. So here was the just the number of bases, the presence of bases, the structure that differs. Here, the G8 molecule, that is the main attacking point. Okay, so this was the basic idea about the hairpin ribozyme. Now we have the hammerhead ribozyme. So the next small ribozyme is the hammerhead ribozyme. So what happens when these RNA molecules basically catalyze the reversible site specific cleavage? Okay, and they cleave the RNA molecule, as well as then the joining reactions within the RNA molecules are done with the help of these RNA molecules only. Okay, and these are basically found in the insects and plants, and it is like named after its structure, that is the hammerhead. Okay, so this is the basic diagram of the hammerhead uh, molecules, and what it does is it basically cleaves itself and then helps in the joining of the self RNA molecule. Okay, and it can cleave the trans also. So what is the meaning of cis and trans? So in cis form, it basically cleaves within the same uh, cell, and in trans means it can target any other, mm, like in other cells also. If we add a nucleic acid in a cell, so that it can act in trans or in that cell also. Okay, so this is the basic difference between the cis and trans. So talking about the cis cleavage, so what happens, this is a basic structure of the hammerhead ribozyme, and this is the cleavage site. So as you can see, this loop is formed, and this is the cleavage within the RNA molecule itself. So here is the cleavage site, and this is the basic catalytic unit. Okay, and this is called as the cis cleavage. And in trans cleavage, when we have this complementary base pairing with the other RNA molecules also, so it will bind to those RNA substrates, and it will cleave the site, and it will not like the function of the RNA will be stopped. So this is called as the trans cleavage. So this is the basic idea about the hammerhead ribozyme and how it is in like it works inside different systems. Okay, so this was about the hammerhead ribozyme. Now the third example is the hepatitis delta virus ribozyme. Okay, or the HDV ribozyme that we'll talk about. So talking about the HDV first, so this particular RNA virus is a co-virus with hepatitis B. Okay, so what happens? This virus do not have the capacity to infect the virus. So it requires a co-virus so that it will infect this particular this virus will infect along with this particular virus. Okay, so for example, this is a liver cell that is already been infected by the HBV virus. Now it has more chances that HDV virus will also infect. Okay, but if the HBV virus is not there, then HDV virus cannot infect itself to this particular cell. This was the first aspect. Now the second aspect is also comes. The literature shows that once the cell is infected prior to the HBV virus, that was long back infection, but then HDV is also present inside the cell, and that is in dormant condition. And this leads to when it basically this particular cell is infected with the HBV virus, this HDV virus becomes active, and it like activates the infectivity process inside the cell. Okay, so it requires HBV virus for co-infection because it cannot infect any other cell due to its nature. And it is a faster type of ribozyme because it like catalyzes 52 reactions per minute, and it is 100-fold faster than the hammerhead ribozymes that we talked about just now, and it is the fastest known self-cleaving RNA. Okay, so this is the comparison between the hepatitis delta virus and the hammerhead ribozyme. Okay, so it also has the same uh, this is the structure of the this particular hepatitis delta virus ribozyme, and this is the cleavage site. Okay, and there are different bases that are conserved, and that depends upon the virus to virus that is being infected. Okay, so this was the basic idea about the hepatitis delta virus. Now talking about the ribosome, that is the peptidase transferase that we have. Okay, so the next example is the ribosome that have the peptidase transferase. So just a brief introduction about the translation process. So what happens? This is the ribosome, large subunit, small subunit. It has three sites, three hypothetical sites that we talk about: A site, P site, and E site. So A site is the acceptor tRNA site. So here the codons are there, three triplet codons, and this site the amino acid comes and binds. Okay, the tRNA with the amino acid charged comes and binds. Then here is the site where the peptide chain is growing, and here is the exit site that has the empty tRNA and that leaves the ribosome. Okay, so what happens? This particular amino acid is needs to be bonded with this particular amino acid. So what happens? This amino acid is transferred on this particular amino acid, and then in the next step, this whole chain shifts to the P site. Okay, so A means acceptor, E means exit, and P means the peptide bond formation site. Okay, so this peptide bond formation is being governed by the peptidide transferase. Okay, so here we'll see a bit of the mechanism that how peptidide transferase helps in the formation of the peptide bond. So here is the crystal structure of the amino acids and how the thing is formed. So this is the this part is from the tRNA. Okay, similar with this. And here at the 3 prime end forms the amino acid. Okay, so yeah, so talking about the molecular mechanism, so this is the A site and this is the P site. And here in the A site at the 3 prime OH, the amino acid is joined. Okay, similar with the P site amino acid, P site tRNA with the amino acid. Now, this particular like this P site, what happens? The amino group. So this the peptidine transferase helps in the breakage of this particular bond and the formation of this particular bond. So first, the 2 prime OH, okay, that is the catalytic OH of the RNA. It basically targets this amino group. Okay, so this amino group then activates and it attacks the nucleophilic attack on the COO bond that is attached to the 3 prime end of the other uh, tRNA. Okay, so this leads to the cleavage of this particular bond and the formation of the peptide bond that is the CONH bond. Okay, so here the peptidyl transferase activates this particular OH so that it attacks on this amino group of the acceptor RNA, which could in turn activate the 3 prime joined amino acid of this particular P site. Okay, so this is the basic mechanism and how it happens. So like this is the intermediate step where it is this end is joined to both this particular oxygen as well as this particular carbon. Okay, so this leads to the formation of the peptide bond like this. So here comes the peptidyl transferase. So here its substrate is basically the amino acids. Okay, so moving on to the application of ribozymes, as I have told you before, also the ribozymes have a recognizing sequence. Okay, that recognizes the target mRNA. This recognizing sequence can be modified for any therapy or treatment so that the target mRNA, being it for any treatment, can be degraded. Okay, so here we'll be using this particular approach to develop therapy that are in clinical trials basically for two things. First is cancer and the other is the hepatitis C virus. So what we do in cancer is, so we have an product called as angiozyme. This targets the VEGF receptor FLT1 mRNA and that degrades that particular mRNA, leading to the stoppage of the functioning of the VEGF. Okay, so first we'll discuss what is VEGF and then the process and how it's involved. Okay, so VEGF is basically vascular endothelial growth factor and that helps in formation of blood vessels. Okay, so why do cancer cells need this particular VEGF, and why it is upregulated in different types of cancer? So what happens? This is a tumor cell, and when it needs to be metastasized, it needs to have its own blood vessels so that the cancer cells can uh, leave through these blood vessels through blood to different parts of the body, through different tissues. And then what happens? These blood vessels need to be formed, and these blood vessels are formed with the help of a molecule called as VEGF. Okay, so this VEGF helps in the formation of these blood vessels, and this leads to different tissues for nutrients as well as from for transferring its own cells, leading to metastasis. Okay, and the process of formation of these blood vessels is called as angiogenesis. Okay, many factors are involved. I am just started with the basic thing. And in most of the cancers, in basically solid tumors, these VEGF molecules are very much upregulated. Okay, because of the formation of the blood vessels. So there is like in cancer treatment, down regulating these VEGF molecules is a very much promising approach that have been found by various scientists. So the scientists decided to target the VEGF receptors. So VEGF, if it has to work, it will work through receptors that are present outside the cell, on the surface of the cell rather. Okay, so what will happen if we design any mRNA vaccine or any ribozyme against these particular receptors? Then that will be degraded, and the function of VEGF will be stopped. So this approach has been applied in this particular way. So what happens? This is the mRNA, the target mRNA, and this is the ribozyme that we have developed for this particular mRNA. It This ribozyme will have complementary sequence or the recognition sequence of this particular mRNA. So it will bind to this RNA, this particular mRNA, and it will cleave this mRNA, leading to that it will not form any protein. Okay, so this the protein is not formed, then the molecule will not be able to function on itself. That will lead to the stoppage of the angiogenesis, and that might decrease the cancer load also. So this was the structure of the ribozyme molecule, and this was the recognition of the target molecule, and this was the cleavage site. Okay, so this was the one of the applications of the ribozyme molecule. The other was the hepatozyme. Okay, not angiozyme, hepatozyme. That basically target highly conserved sequence in the hepatitis C virus mRNA. Okay, so that too in the positive strand mRNA. Okay, so this was the structure like this. Like in the previous one also, we have a recognition sequence that will bind to the target HCV RNA, and this was the cleavage site. So if it will cleave, then no infection. Okay, so these two, hepatozyme as well as angiozyme, are in clinical trials currently, and this leads to the end of today's lecture, where we have discussed different type of ribozymes and how these functions inside the cell. And if you have any questions, you can ask in the next class. Okay, so thank you for the session.